All right, thanks very much for inviting me here, and very happy to be here. Me and Catherine are here together, and originally from Ireland, and now we've lived in Boston for all our lives since then. But anyway, I'll be talking about manganese overexpression because it's a very, very sort of forgotten about metallotoxin, which is closely linked to Parkinson's disease, Parkinson's. And so we've done a lot of work with the help of some investigators in recent years. And so manganese overexposures in MGH, we did the work. Manganese overexposures can interfere, I'll show you more or less, can interfere with the pathways of neuroprotection. And so what I'll show you is that, in fact, the manganese overexposure is abrogating APP, which is the amyloid precursor protein, and stopping translation of this as a neuroprotectant. Most people regard APP as a precursor to amyloid in Alzheimer's disease, but people forget that under the age of 40, no one's going to get any amyloidosis, and they're the people who need to have the protective effects of APP. And historically, we've always worked with ferritin, which is the iron storage protein. So the talk very much centers around iron homeostasis, because APP we published in Cell JBC, various places like that, is linked to iron homeostasis. So manganese encephalopathy is unique pathology. It has got symptoms of similar to Parkinson's, where people become their gait is screwed up and they become ataxic, whereas the psychological symptoms are also present. You have compulsive behaviors, running, fighting, singing, liver cirrhosis, and all various kinds of disturbances. However, manganese neurotoxicity is also linked to a sort of brain hepatic encephalopathy, similar kind of response that you get in the liver with manganese. So basically what I'll show you is the data where we're able to have done bioinformatic analysis of the manganese toxicity pathways and come up with a simple therapy to manganese overexposure. And that's the case of use of purines, inosine, as a simple sort of curative mechanism. So what I'll be showing you is that APPS is neuroprotective. Manganese abrogates APP translation and causes neurotoxicity, whereas urate jumps in and restores APP translation, and it's correlated to neuronal survival and protection from this movement disorder. I thought I'd just to put in two colleagues who recently memoriam who worked so heavily on iron homeostasis, Barry Poor and Marian Wessling-Resnick, who unfortunately died recently, and their loss is greatly felt. But they were the people who first showed how iron comes into the body. And Marianne in particular was very involved in showing how manganese interfered with iron transport in the body. Iron transports into the body basically through proteins on the lumen of the gut, which are DMT1, which then transcytoses to ferroportin, which exudates the iron from transferrin from the diet into transferrin in the blood. And I'll show you that APP, we discovered, is involved in this process in the neuron by helping to pump this export process of iron, too much iron is toxic, out of cells. And manganese actually promotes this iron buildup, whereas urate, I'll show you, through the APP pathway binding to transferrin, basically rescues the whole thing. So basically, APP and ferritin. Ferritin stores iron, APP exports iron. Too much iron is oxidatively stressful. And the genes that control iron homeostasis are through the translation, not transcription pathways, not transcription, translation, where RNA binding proteins bind to iron responsive elements, which we discovered in the amyloid precursor protein, traditionally thought of as a pro-amyloid, but now with Ashley Bush and colleagues have shown that it's an iron export protein. And we published since in our original papers, 2010, we published in Journal of Neurochemistry a few times, which I'll show you the data for. I'll keep moving forward. And so basically, ferritin stores all your body's iron. It's translationally controlled by iron homeostasis through these iron regulatory proteins, which come on and off with iron. Iron removes them, allows translation to occur. Then you get ferritin to store all that nasty oxidative iron. Hamish Monroe was the leader in this area decades ago, as was Klausner and Tracy Rowell in the NIH. We contributed by showing that APP has the same 5' untranslated region control to iron regulatory proteins. And indeed, with Ashley Bush, we showed that APP protein is in an iron export protein. And here with Catherine Cahill, we showed that the RNA stem loop in the APP 5' untranslated region controls this iron homeostasis. That's published in JBC. So I'm just kind of giving you an overview of the regulation of iron homeostasis, which we show that APP contributes to. And iron regulatory protein 1, the favorite of the likes of Liz Thiel, Bill Walton, and Rick Eidenstein, binds to IREs and it represses them. Ferritin and APP are two examples of that in iron deficit. Iron surplus, they go like gangbusters again. They get translated to try to store that oxidative, ferroxidative iron in the cell. And here's Ashley's original picture, where we published that APP binds to ferroportin and is involved in iron efflux from cells. And I'll show you some data in Journal of Neurochemistry in 2018 and more recently in Biomolecules this year with Catherine and Sanjan Sarang, backing this up fully. APP is an iron exporter. And so you see APP is the protein on all membranes in all cells, but very much focus of neurologists in the membranous regions of neurons because it makes the amyloid plaques by cleavage events of base and gamma secretase, which I could go into for hours about, because a huge number of man-hours have gone to talk about amyloidosis in Alzheimer's disease. We, however, focused on people under 40 years old who have no chance of getting Alzheimer's disease. And the APPS, originally shown by Saito and his colleagues in San Diego, is neuroprotective. We back that up very well in our recent papers. A neuroprotective against iron damage, APPS. So it's very, very protective. And you can see that, so you can either get cleavage to amyloid or protection from APPS, which is 90 kilodaltons. So I'm just showing you the alternative pathways of APP. Now, so over the years, we've tried the therapies where we've tried to do what everybody else does, anti-amyloid therapies against Alzheimer's disease, and much criticism, a lot of money, a lot of petrodollars and all that sort of stuff, which we've contributed to. And so if you knock down APP translation, you knock down amyloid. Well, why not just look at it from a slightly different viewpoint and see the neuroprotective side of APPS? And lo and behold, you have this disease called manganism, where to make a complex case simple, manganism is like Parkinson's, but as any reviewer on an NIH grant will tell you, it's not Parkinson, because it centers on the globus pallidus in the midbrain instead of the substantia nigra. Symptoms are irritability, difficulty walking, speech disturbances, compulsive behavior, running, fighting, singing, liver cirrhosis. So I pulled down those symptoms from the internet, but you can see that the bottom line behind this talk will be that neuroprotective strategies against manganese-induced toxicity through APP and ferritin destroying their activity can be abrogated by simply applying uric acid or urate to cells. And that's in the paper this year with Catherine and Sanjan Sarang. And here you see some of the occupational hazards of people in smeltering plants, battery factories in the third world, where people suffer from this manganism, which is a gait disturbance in younger people with a Parkinsonianism. Iron siderosis is also a problem. We published with people, Wei Wu Lu and Fudi Wang, actually a meta-analysis showing manganese not only affects adults work in the occupational smeltering industry, but also children exposed to environmental toxins of manganese overexposure in the water or air around the sites of such places. They work in Zhejiang, China, but we work here in Boston, but Fudi had been a visiting scientist. And we did a lot of meta-analysis, and we're able to show, on average, after extensive review, backwards and forwards with lethal reviewers, that the IQ, general IQ, can be averaged. I know it's a very kind of controversial, but it kind of gives you an idea that children's IQ can be reduced by 2.5 units across different cultures by their estimates from manganese exposures in industry. So that's not me in the Western world trying to claim that. That's people from the third world, or not China, not the third world at all. But so anyway, we have got our model where basically APP is pushed onto the membrane of all cells normally, and it actually binds to the iron efflux protein that I showed you, Marianne Westling-Resnick had worked so much with, and it pumps iron out of cells when it's present in too much excess. We showed in our J and D papers, Journal Neurochemistry, with lead and manganese, and Catherine Cahill was very instrumental. Vivek Venkatanarami was very instrumental, that the manganese can block translation of ferritin and APP and pull neuroprotection away from cells. And that data can be seen in these Western blots, where you can see that manganese reduces APP dose responsively. It eliminates ferritin in SY5Y cells. And you can see that also iron does things in the opposite direction. So the point is that this is not just present in SY5Y cells, it also goes on in mice and rats, as supplied by our collaborators and us, where we did manganese overexposure to mice and rats, and we're able to show that ferritin and APP also goes down in the rodents. So that's all published, and we showed that it goes through the 5' UTR, that the inhibition, it can knock down the 5' UTR of the IRE encoding ferritin and APP. So to cut a long story short, we were able to also, Vivek was able to show that when you overexpress APP, indeed, it protects the cells against manganese death. So too much APP is protective to these cells. So I don't want to get too much into detail, but you can see that there is a protective effect, and it's by taking away the oxidative damage from too much iron buildup in the cells. So the model is that manganese or lead can cause toxicity. This is not the only mechanism. I'm not so naive as to think that manganese and lead only do this, but they do knock down APP and ferritin, and they are protective proteins, and the cells die, and they can be rescued back with too much APP, when you overexpress APP rather, artificially. So then along came Sanjan, and he conducted pathway analysis of SY5Y cells. He's a high school student, works with us, and he's one of these. Thank you. One of these geniuses who that is very, very savvy with computers and bioinformatics and mathematics. And he showed that if you add manganese to SY5 white cells, he pointed out, we repeated what I said, that manganese is a heavy metal which contributes to human toxicity, and is commonly an occupational overexposure problem, quite severe clinically, actually, in younger people when they work in these smeltering plants, occupationally. The goal is to identify the key genes in these pathways and see if we can, I'll cut the story short for 5 minutes, that the key genes we found through Sanjan's good efforts were purines, and urate was the top of those key genes, and urate protected the cells from manganese toxicity by this APP pathway. And so he did this analysis of these microarrays from bioinformatics studies online by a fellow called Gandhi in 2017, and was able to draw all kinds of beautiful volcano plots and etc., where he was able to look at all the induced genes from manganese poisoning in cells, and was able to list, for example, SERTs and nucleotide salvage pathways. That's where purines come in, cleavage of damaged purines and other pathways, which I won't go into now. But the purines indeed came up, and you can see that we were so interested in that. That's why we followed the urate as the cure to manganese poisoning. And the gene changes revealed also metallothionein 3, but they primarily illustrated that purines, if maneuvered around, could be used to offset APP's neurotoxic effect when manganese was inhibiting it. And so we were able to, using targeting, urate has been used for Parkinson's disease in clinical trials, unsuccessful. However, we have used it for manganese poisoning. And I just want to point out that when we used it in pathways, which we just published recently, we were able to show that urate could restore APP, although it left ferritin the same with manganese inhibiting it. So what we discovered was that indeed urate could recover a lot of manganese toxicity, but primarily it appears to be through APP. And what we have found is that, what we believe is that the fact that urate restores APP under conditions of manganese toxicity is a contributor to its uses as a therapy for manganism. And I'll just wrap up there by just pointing out that as well as urate, there are other mechanisms which we have found which actually restore ferritin as well, but I won't go into those details because I think the urate story is simple and worth remembering as a pathway which we published this year to offset this terrible affliction by taking inosine as a supplement, which is the precursor to urate in affected individuals with manganese poisoning. So I'll finish up there by skipping through the slides of the small molecules, BL1 in particular, which restore ferritin, but focus instead on just the future directions which we're partaking of in our pathway to help people with manganese poisoning and lead poisoning. And there is the team that we had the core work with. Catherine and myself went to the Medical Education Center with Yunsi, Suresh, and Henry and Harry, who did a lot of the very good work with us. And so more or less that's the story so far.
Questions?